Hello, and thank you for listening to my presentation on the influence of fatigue on forearm flexors and extensors during isometric contraction. Voluntary isometric muscular contraction can be measured with the use of surface electromyography, or EMG. The peaks shown in the EMG readout represent the motor unit recruitment occurring in the muscle. When fatigue begins to set in, the amplitude of the EMG peaks typically increase because more motor units are recruited to maintain force output. However, contradicting research has been done that suggests that EMG amplitude is not a good measure of motor unit recruitment, and sometimes the amplitude will decrease with fatigue. The purpose of this study was to see how the amplitudes of the EMG changed as the flexors and extensors of the forearm muscles fatigued during prolonged isometric contraction using a hand grip dynamometer. The hypothesis was that the amplitudes would increase as fatigue increased with prolonged contraction. Subjects for this study were recruited from the Biomechanics Lab for the Regis University Doctor of Physical Therapy class of 2019. 8 total subjects were recruited, 2 females and 6 males. The subjects were first prepped for electrode placement. The muscle bellies for the flexors and extensors were palpated, and those sites were shaved and swabbed with alcohol. The EMG electrodes were then placed in parallel to each other and running in the same direction as the muscle fibers. After electrode placement was confirmed, the hand grip dynamometer was adjusted to fit the subject's hand. The subject performed a maximal voluntary contraction, or MVC, on the dynamometer from a stainer position. This value was recorded, and 25% of this value was calculated. The subject then held this 25% MVC value in a stainer position until either fatigue or 300 seconds was reached. Percent MVC and force were recorded at 15-second intervals. The results obtained showed a decrease in percent MVC as time went on. This is depicted in Figure 1. The red line represents the percent MVC and error for the flexors, and the blue line represents the percent MVC and error for the extensors. Both of these data sets show a downward trend. Figure 2 shows force percent standard over time. These values remained relatively constant, showing that the participants were able to maintain close to 25% of their MVC. This data did not support the hypothesis. An increase in amplitude was expected, but a decrease occurred, despite the fact that overall force output remained fairly constant between 22 and 25% MVC. There was also a low correlation between the data points for both extensors and flexors compared to percent MVC. The decrease in amplitude and low correlation may have been due to lack of experience using the software, imprecise electrode placement, or small sample size. However, other literature has shown a decrease in amplitude during fatigue and a lack of consistent association between amplitude and motor unit recruitment. Therefore, more research may need to be done in this area. In terms of clinical relevance, this study demonstrates the importance of knowing how to read and interpret EMG data. Knowing how EMG data can indicate fatigue can help in both preventing injury and prescribing appropriate levels of exercise to keep a patient from fatiguing too drastically or too early in the exercise. Thank you for listening.